relatively concentrated perspective on the 201 class. Some of you took the 101 course when it had a bunch of machine learning in it, and some of you probably haven't taken it with the machine learning component. So what we're going to do in this class today is cover some of that. We're going to go over some of that machine learning material again in some depth. So for some of you that might be partially review. So I'm a research scientist at this nonprofit called the Child Mind Institute, and I study the mostly fMRI, how the brain networks are organized in fMRI, mostly focusing on these sort of interactions between these cortical and subcortical regions. Let me see if I can annotate this. How about this? So can you guys see this red mark? I guess probably can. In the bottom right-hand corner here, or let me just draw maybe with this. So just talking about this part over here. So the systems that I study are specifically called corticostriatal circuits, and they are kind of the circuits of the brain that are responsible for a lot of skill learning and like habit formation. And one of the reasons why they're so interesting is they're not only really closely related to learning, but they also are one of the circuits that you can target in most successfully in deep brain stimulation. So we don't do a lot of like cyborg stuff yet in neurotechnology, but deep brain stimulation and cochlear implants are two exceptions to that. Cochlear implants being implants into your into your ears that can help you hear. They're basically like little microphones that relay the information from the that they're receiving from the world and send that as neural signals into the brain, and people can hear as a result of that. Deep brain stimulation is often used to put electrodes into deeper parts of the brain, such as the thalamus, which is a part of the brain that I study, and can be used to stimulate those deeper brain regions to sort of correct for chemical imbalances that may happen, for example, in the case of Parkinson's disease. So I'm going to, okay. Oh, I can't, I need to get rid of that annotation. How do I do that? Clear. Please bear with me while I learn how to navigate, learn how to navigate this. Okay, great. Okay, so today we're gonna focus mostly on some neuroscientific topics and machine learning. And whereas before in the like original Neurohacking 101 that had the 1 day of neuroscience and 1 day of machine learning stuff, we went into some of the topics in machine learning kind of in a little bit more of a cursory manner, just because there was so much material, it was a little difficult to cover everything. But what we're going to do today is we're going to what I'm going to do is I'm going to talk to you guys about a particular way of thinking about fMRI data and how that kind of lends itself really well as a lens through which we can understand a lot of kind of concepts, essential concepts in machine learning. So the two Python packages that we're going to be learning about today are Scikit Learn and Nilearn. While we're on this, could you guys use the reactions? If you click the participants tab, could you tell me yes or no if you've installed Anaconda and have it set up on your computer? So I will do it like yes, like that.
Yes is thumbs up.
Yes is, yeah. If you click the it's not the reactions tab, but the participants tab. In the bottom,
Okay, I see it.
yeah, yeah. How about you, Campbell? Oh, I see, you're green. Okay, got it. Um, uh, Jamil, could you unmute for a second?
Yeah,
What's your Python setup like?
I just use pipenv and Python 3. And I'm
Okay, so do you have Scikit Learn and Nilearn already set up and installed?
I do. I don't have Nilearn. I'll do that right now. I have Scikit Learn.
So the ones that I'll just type into the chat to make sure you guys pip install these. So it'll be pip. You want to install Nilearn. And mglearn. And mglearn is a machine learning library that's specifically designed for didactic purposes. It was created by Andreas Muller, who's a really great neuroscientist and computer scientist at Columbia. And we're going to be working through some of his material in the machine learning review. Okay, so let me move this back.
Is there like a test command or a test notebook that we can run just to make sure we have it installed properly?
If you, I didn't create a test notebook like I did last time. If you open up a Jupyter notebook and you can import them, you should be good. Okay, so machine learning is a really kind of exhaustive field. Scikit-learn has this beautiful algorithm cheat sheet that I know I've shown you guys before, where you can kind of think about what question you have, maybe starting with a data set, and then go from there, answering yes and no questions to find out which sorts of algorithms you should have a look at. And if you look up this Scikit-learn algorithm cheat sheet, you'll find like a web page where all of these cells are actually links. So for example, say I want to do some kind of simple classification. One of the first classifiers that we're gonna review today is called k-nearest neighbors classifier. So say, okay, I start here, I have more than 50 samples and I'm predicting a category, and I do have labeled data, and I have less than 100,000 samples. I can do singular, or I can do linear square vector classification, which we will talk about, but if more simply, I can do k-nearest neighbors, which we will discuss how that works. So that's kind of how this sort of flow chart works. Just as a review of last time, what we did is, I introduced everyone sort of really bare bones how the Neurohacking 101 worked, right? Is that you could take a Python bootcamp with it. So there was like a 4-hour Python bootcamp where we would introduce people to Python and then teach them how to make things like these basic plots through these simple exercises. We discussed sort of the like international online hackathon community, specifically this one called brainhack.org, where we organize these international brain hacks where people with hacking skills, not necessarily neuroscientists, come together and get to build cool tools and hack together on different projects. In this era of sort of new collaboration that's going on with, with COVID and everything, this has accelerated pretty dramatically. So now, like, there are conferences that are happening like on a basically kind of like a weekly basis. It's pretty crazy. So scientists like are organizing on Twitter and all meeting all the time, mostly for drinks, I think, but like they're also, you know, talking and discussing work and stuff like that. So the science Twitter world has kind of like cranked up its speed quite a bit. And so there's this open science movement that I'm like, I'm very invested in. I'm very excited about. And Python is really one of the keystones of this open science movement. We discussed this last time. There's a lot of different kinds of packages that are designed to handle different types of scientific data. Like, for example, we have NiPy for neuroimaging data. AstroPy has a lot of different kinds of specialty functions and packages for astronomy. BioPython is like for the biological sciences, etc. And one of the cool things is that they all kind of work together and share a lot of the same underlying architecture, right? So if you're familiar with how some of the functions work in NiPy or in Nilearn, you'll see that under the hood, they're running actually the same NumPy, SciPy, and Scikit Learn functions that AstroPy and BioPython, etc., are all using. So with this open science movement, what's one kind of exciting thing that's happening is that as advances are made in one field, they sort of start having ricochet or trickle down effects, like real trickle down effects on other fields. And so we sort of all benefit from this. So a good example of this, for example, is Scikit Learn. This is a machine learning a machine learning Python package that we have discussed in the last class. And a lot of the creators of this, of Scikit Learn, are also contributing to creating neuroimaging specific versions of the things that they've created for Scikit Learn. And we're going to go a lot in depth in reviewing all of that. And as a sort of required plug, I always make sure that I tell my students about Sci-Hub. Sci-Hub is a place where you can get basically all papers that have been published. You can get them downloaded to your computer without needing library access. Yeah, Jamil?
I was just wondering if any neuroscience work happens in R, or if there is any community in R around open neuroscience.
There is definitely an R-based an R community for neuroscience in the sort of neuroimaging specific community of neuroscience. It's smaller than the Python-based community. Still, I would say, like, of all the languages, I would say probably MATLAB is still one of the dominant languages in neuroscience, neuroimaging in particular, just because MATLAB has just such a first mover advantage. It has so many specialized engineering and digital signal processing type libraries that it still has quite and a big pocketbook. It has a big advantage over R and Python. But I'll just briefly say a couple of reasons why MATLAB Python would be preferable to MATLAB. So MATLAB has I would say probably primarily is MATLAB has this licensing issue, which makes it so that every time that you run a proprietary function of MATLAB, it has to check that your license is still working. And so you might, for example, have some kind of internet delay or connection issue or something like that, and as a result of that, your analysis might fail. Not having anything to do with your code not being right, just has to do with this proprietary element of how MATLAB works. MATLAB's also fast for some things and slow for some other things. And I'm not the computer scientist to really go into this, but from what I understand, not all of MATLAB is compiled in C. Some of it is, some of it isn't. And the stuff that's not compiled in C is kind of slow. But there are things in Python that are slow too. So it's not necessarily that Python is a lot faster. Okay, I'm gonna keep going. So we also covered some things about last time about sort of brain networks, both structural brain networks and functional brain networks, and how we can construct them. In the case of functional brain networks, from taking the time series from different parts of the brain, and then correlating them together to get these kind of like correlation-based graphs. Had you guys do exercises where we were grabbing different seeds and different voxels of the brain, and then we were extracting time series, and then we were pre-processing the time series with different amounts of frequency, different frequency bands to kind of see what different kinds of signals there are. And then another thing that we went over is these online machine learning repositories that you can have a look at. So the two that we used in the last one, and we will use this breast cancer one again today, are in this University of California, Irvine website, where they host thousands and thousands of different kinds of machine learning data repositories, where if you're learning for the first time how to do scikit learn, you've done some support vector machines for the first time on some tutorial like what we're doing today, and you get familiar with what the data structure looks like, etc., to get to the point where you've gotten to the point where you actually understand the workflow. You can now go and take data from here and plug it in and see how that works as well. And they have the data on here organized in such a way that they'll tell you like, oh, this is good for classification, this is good for regression, etc. Okay, so I'm gonna do a little overview right now of what we're gonna talk about today. So the first thing that we're gonna do is we're gonna do some review of MRI. I'm gonna go a little bit more in depth about how MRI works than I did last time, just because it's such a freaking cool technology. I would love to go even more in depth with it. Maybe in 202 or 301 we can do something like that. And then we're gonna have a little Q&A and just see what kind of confusion points or sticking points you guys are having with the material, or just curiosities really. I think since we have a smaller class, we can have a little bit more time devoted to Q&A, especially since I think we spent about like an hour in the 101 course just setting up people's computers. And for the most part, we should be pretty much good on all of that. And then I'm going to talk about this idea of decoding. So decoding was kind of like the first, one of the first like machine learning perspectives on fMRI data. And James Haxby, who's up at Dartmouth, is the one who came up with this, and it's a pretty cool idea. And then I'm gonna kind of introduce you guys to the idea broadly, and then with the first two Jupyter notebooks that we're gonna work on, which you guys should all have downloaded, we're just gonna go over sort of like the high level understanding of how that study worked. And then Randy, you have a technical question? Do you want to? You're unmuted now. You want to say?
Yeah. So I was trying to pip install MGlearn, and I get an error. Package image.io requires a different Python 2.717. I think I'm on 3.7.
Huh. Okay. Try.
But I may be able to switch the environment.
Try specifying the version of MGlearn, because if you're on Python 3, then I just typed it in the chat. If you try to do MGlearn, I think maybe without the spaces actually. So like,
Okay.
Yeah, you also might need to uninstall, but typically when you upgrade, it will do the uninstallation during that process. So maybe try that, Randy.
Okay.
What I just typed in, and let's see if that works. I think that the technical challenges of the of debugging other people's computers over Zoom is a thing that I really don't know how well it's going to work out.
Yeah, no, it doesn't like that either.
Yeah, you might like what Drew Mills said. You might want to uninstall. So it would be pip uninstall. Another thing that you can try is to use the user version of the installation. Yeah, and if you want to paste the error into the chat, that would be good.
All right, trying it again. Yeah, no, it's still not liking it. There's no matching distribution for MGLearn. Did I type that in right? Oh, I'm sorry, I typed it in wrong. Need an extra dot. Ah, there, seems to be going through.
All right, awesome.
Thank you.
Yep.
Oh,
Okay.
no, it's still burping on same.
Did you?
I think it wants Python 2.12717.
That's strange.
Unless, yeah.
I run Python 3 and MGLearn works fine on Python 3 on my computer.
Just to make sure that you're installing it with the right Python, you might want to try pip3 install.
Okay.
Yeah, if you have Python, yeah.
Okay. Okay, that's maybe that looks like it's working.
Thank you, Jamil.
Thank you.
Yep.
As always, the debugging relies on a community effort, right?
Yep.
So I've gotten thankful and used to that. Okay, so let me know if you guys have any other issues or questions or anything like that as we go on. So after we cover the first couple notebooks, what we're going to do is we're going to go into sort of like after the Haxby sort of changed people's frame of reference with regards to how to handle fMRI data from this like multivariate sort of more machine learning perspective. After that happened, then Nicholas Kriegeskorte came along and created this thing called the Searchlight, which we're going to discuss. And people started looking at fMRI from a really different perspective, kind of less from a neurological, psychological perspective, and more from like a machine learning information theory type perspective, which led to, in 2004, 2005, the proliferation of a lot of different kinds of machine learning pipelines. And then we're going to go into this notebook, Jupyter notebook, that kind of covers how searchlights work a little bit, and an example of what the results look like. And then we're going to see what the results of the Searchlight look like compared to what we were looking at before. The Searchlight is going to be our first time that we're going to use something that's kind of like traditionally machine learning, which will be the support vector classification. So we're not going to talk about it much in that notebook, but in the next section, which is the machine learning primer, we're going to go into more depth in all of the machine learning issues. So the machine learning primer is kind of like the midpoint or so of the course, give or take. I I think the key point here is that since I don't have like a whiteboard, and since you know, how we would talk about things was a little bit more fluid when we're in person, what I've done here is to explain certain like essential concepts. I've just grabbed short clips of videos that we can watch during the machine learning primer. And then we'll be doing Q&A sort of as we go on, as we go through sort of talking about these different questions. And then we are gonna talk about, have a few more, do a few more notebooks, and that kind of cover this. So like now that we understand the more advanced machine learning stuff, we're gonna talk a little bit more about how more advanced versions of this decoding thing can work. In particular, we're going to look at the in the masks notebook. We're just going to look at like where in the brain these types of information is mostly held. In the different estimators notebook, we're going to look at what's the effect of using different kinds of machine learning algorithms on the predictions. And then in the GraphNet notebook, we're going to kind of cover some of the ideas about regularization and penalties that we talked about in the machine learning primer. And then at the end, if we have time, I'd like to maybe talk a little bit about some like more future-facing technologies that are coming out in neuroscience that I think are pretty exciting. And since we have such a small class, and I think we should just feel free to ask questions throughout the whole thing. I'm going to be having specific times for Q&A, but I want you guys to just feel free to just just type questions right into the chat at any time, and we can just cover them as we go. Okay. So with that, let's open up for questions now. Does anyone have any questions about the organization of the day and how we're going to structure things? We're going to try and take like a bathroom break like every hour or two, 5 to 10 minutes or so. Get some coffee, get some tea, etc. And then at some point we will kind of collectively vote on when we want to do lunch. So at GenSpace, we would take some. So here is sort of the beginning of the MRI physics talk. So the first thing we need to do when we discuss MRI and how it works is talk about the physics of how MRI actually works, because that's a really key component to understanding how we're probing these physical systems. Everyone can still see my screen share, yeah? Okay. Just, is that let me get a let me see if I can get this. It says from my it says from my perspective here that, so I'm off screen looking at like other materials too. It says that you guys can't see my screen. Is that true, or can you see it? If you give me a thumbs up if you can see my shared screen.
You can see the MRI scanner cut away, but sometimes when you're clicking something, there's like a white box that covers up part of the slide, or something that looks a little funny. So that's what it looks like.
But right now, at least?
Yeah, now it's
okay. Okay. So for some reason, it says that it says that sharing is paused right now on my screen. So, okay. Good. Glad that's working. Okay. So, right. So the first thing that we're going to talk about is MRI physics. So, okay. So atoms. Every atom, you can see like this diagram of the atom here. Every atom has this characteristic called a spin. I call it a characteristic because this spin, the idea of spin, like something spinning, is all like a classical mechanics perspective. Actually, like atoms they're not spinning in this sort of like idea that we have. But for the purposes of MRI physics, the cool thing is that basically all of it is classical mechanics. So we don't need to think about like quantum physics or anything like that. Mostly we can just think about atoms actually physically spinning around, okay? And so you can think about the spin of the atoms as having an orientation, like which direction it's going, and a frequency of the spinning, like how fast it's spinning around, right? How quickly it's spinning. So the cool thing about this is like what chemists and physicists discovered in the 1920s is that for a handful of atoms, they exhibit this really remarkable behavior. And so what they were able to do, the physicists and chemists, they were able to measure the frequency at which this atom was spinning, right? So how quickly it's spinning around. And they found out that if you send a magnetic pulse that's at the same rotating frequency as this atom, when you turn the pulse off, the atom will send you back energy. So it absorbs some energy. If you send it at the same, if you send a magnetic pulse at the same frequency as the rotational frequency of the atom, this is called the Lamour frequency. If you send it at the same rotation, you can transfer energy from the radio frequency pulse into the atom. And then when you turn it off, that energy will be sent back out, right? And it doesn't really, and that phenomenon, that is called magnetic resonance. That's the idea of magnetic resonance, the resonating of the atom after you have pulsed it with this rotating magnetic field. And it doesn't take a really strong magnetic field. It's just a pretty weak one, actually, to get this behavior from atoms. And so luckily for us, the atoms that show this characteristic really interesting behavior just happen to be all the same atoms that we are made of, right? So like hydrogen, oxygen, carbon, and phosphorus. These are like essential building blocks of life, and they all happen to be ones that exhibit magnetic resonance. So nature, is pretty tricky in a lot of ways. We don't really understand how it works, etc. But like in some particularly strange cases, nature kind of like lays out the red carpet for us in a way. And it doesn't happen very often. So it's a pretty big deal, right? So like of all the atoms that you observe in the periodic table, the only ones that have this remarkable ability are the ones that we happen to be made of. It's a really like amazing feat of nature, but also the fact that we can probe this type of thing with technology is just absolutely is wild, really, really wild. So hydrogen and carbon and phosphorus are the ones that we mostly use for imaging. And for all of the MRI that we're going to talk about today, hydrogen is going to be the type of imaging that we are going to be talking about. And so the signals of how that resonance looks like is going to be different depending on which atom the molecule is in. So not only can we measure atoms, we can not only measure the atoms inside of things, but we can also identify which molecules are inside of things, because every molecule, right? So water, for example, 1 oxygen, 2 hydrogen. The hydrogens on the water molecule have a very distinct resonance profile, right? And every different molecule, those hydrogens will have a different kind of resonance profile. And that has to do with basically how much of the electron cloud from the hydrogens are getting pulled into these other atoms, right? And so there's this cool technique, which was kind of the very first technique that came out in MRI, called magnetic resonance spectroscopy, where you could use the magnet to actually figure out how much of what molecule is inside of the tissue that you're looking at. And the cool thing is that you can use this exact same technology in the brain as well. And in 2016, I published a paper using magnetic resonance spectroscopy to look at one of the major macromolecules in the brain called N-acetyl aspartate, and to look at it. It's involved in basically it's a metabolic marker in gray matter and white matter, and to look at how its distribution and individual differences in the distribution of this metabolic marker were related to intelligence, which I can link to. We can talk about that paper later if you want during Q&A or whatever. Okay, so not only can we use the MRI to get a sense of, you know, which atoms are in, but we can also get a sense of like the actual molecules and the chemical makeup as well. Okay, so atoms, right, have spin, but our bodies aren't like crystals or metals, right? So like our atoms are moving around all the time. They're bumping into each other, and overall they don't have actually a total direction for the sum of all the directions that they're spinning in. So we use the you know, this big MRI machine. The point of the big MRI machine magnet is actually just to create a homogeneous magnetic field. So that's what we're actually doing there. And what we create with this magnetic field can be tens of thousands of times as strong as the Earth's magnetic field. And anything that goes inside of that magnet aligns parallel or opposite to the magnetic field, like we're seeing, right? But luckily, there's a slight difference in the number of atoms that are aligned in the magnetic north as in the magnetic south, because one is a slightly higher energy state. So, so the lower energy state is the preferred one. And the higher the strength of the magnetic field, the greater the difference between this up and the down, right? So all of the signal that we get from the MRI, every bit of the signal that we get from the MRI is just the like 3 parts per million more that atoms are pointing up versus down, right? So it's like 100th or 1,000th of a percentage of the total signal that we get, right? Because that small little energy difference, that's all that we're actually picking up in MRI. But the stronger that the magnet is, the more of that energy difference we see. So just to give you guys a sense of how strong these magnets are, you guys may have heard of the Large Hadron Collider. This is probably the most expensive scientific project ever conceived. I could be wrong on that, but I'd guess it's somewhere up there. So they send particles of like very like heavy atoms like gold and other atoms, lighter atoms like hydrogen, et cetera, at like a fraction less than the speed of light, 99.9, et cetera, percent the speed of light. And they accelerate it very rapidly. And in order to get it to conform to those, to that ring that they send it in, they have these strong magnets all around the edges, right? The MRI machine that we put ourselves into and that we do for, that is like totally clinically safe, et cetera, they are at 3 Tesla. Those machines are also at 3 Tesla. So they're the exact same strength of magnets that we use in Large Hadron Collider as what we use in all of the stuff that we're going to talk about today. Okay, all right. So to and as always, if like throughout this, you guys just raise a hand or let me know you have a question, et cetera, and I'll answer it. Unfortunately, it doesn't always show me who's raising questions, so yeah, I guess I'll tab over just to make sure. How many Tesla is the Earth's magnetic field for comparison? So our that's a good question. I need to do the division, but basically ours is the 3 Tesla is 60,000 times the strength of the Earth's magnetic field. The cool thing about this though is that, so like I said earlier, the magnetic field that you use to actually get the signal out of the material is very weak, right? So you actually don't need a very strong magnet. If you are okay with that ratio of up to down atoms being very, very small, you can actually use the magnetic field of the Earth itself as the magnetic north. And they have these things called low field MRI machines. And you can actually get like a kind of sort of image out of it. We did this in grad school where we'd put an orange inside and then we'd image it, and then you would see that it's a circle. You'd see something, but you couldn't see in it or anything like that. You would just see like what it was on the outside. Okay, so to recap, so we go in the MRI scanner, all of our atoms are aligned to the main magnet's magnetic field, and it's completely safe because the alignment of the atoms, this aligning of the atoms isn't actually strong enough to make any of them break any of their hydrogen bonds or chemical bonds. So atoms still behave as they would normally. And then we apply a very small magnetic field that is rotating at a specific frequency that's the same rotational frequency as the atom that we're interested in. So in this case, it would be hydrogen. So we turn on a spinning magnetic field at the same frequency as hydrogen, and this matched rotation actually causes hydrogen to change its alignment. So from being aligned with the magnetic north of the magnet, now it's aligned perpendicular to the magnet, okay? And this second magnetic field is turned on very briefly. So then the hydrogen atoms begin returning to their original alignment with the main magnetic field. And while they are doing this, they're actually rotating around the magnetic field. So can you guys see can you see me at all? So basically, like, if the main magnetic field is like this and my pen is this atom, after the pulse, or I'll do it the other way. After the pulse, the atom is now at 90 degrees. When the secondary magnetic field turns off, now the atoms now are rotating around this main magnetic field. So whereas first they were rotating like this, now they're rotating like this, right? And as they come back up to the main magnetic field, they're emitting energy the entire time. And we are capturing that energy with the MRI machine. So the MRI machine is not only putting energy into the atoms, it's also receiving them, right? And we record all these signals, and we use differences in the signals based on different places to reconstruct the image. There is a really, really cool element of this and frame of reference of this whole thing that I'm not going to talk about, that I highly recommend you check out, called K-space. So K-space is the frequency space that all of this data is recorded into, and then it is transformed from frequency space into signal, basically, or it goes from signal to frequency and then back to the image. And this transformation, using a technique called the Fourier transform, which you may have heard of before, is how it happens. And I think it's probably one of the coolest parts of MRI, because where you go in frequency space, as in like, there's a one-to-one mapping between how you make those so that secondary magnet, it's called the radio frequency pulse, how you design those radio frequency pulses has a one-to-one correspondence with where you go in K-space. And where you go in K-space has a one-to-one correspondence with what kind of image you get back on the other end. So if I, for example, sample the very center of K-space, what I'm going to get is sort of the large-scale structure of the image that I'm looking at. If I were to sample the outside of K-space, I get the fine detailed structure. And by what like biomedical engineers and people who design these MRI sequences do is they create sequences that are designed to sample K-space in a way that's advantageous for doctors, et cetera, et cetera. So like when a certain MRI image looks a particular way, it's part of that is due to how K-space was sampled, which has a resulting relationship to how these MRI radio frequency pulses were used. Okay, any questions on that before we? What's the use of the low field scanner other than scanning the shape of oranges? There's a really fascinating company that I wouldn't say obsessed with. Maybe it's more like an infatuation. I don't know, it's something like that. They're called Hyperfine. For those of you in Connecticut, they're in Connecticut. And they basically use low field MRI with low field MRI merged with deep learning to create like MRI machines that you can wheel into a patient's bedroom. So it doesn't use like a super heavy superconducting magnet. So you can wheel it around a hospital and get MRI images. So you can actually track the progress of like a surgery day to day, which is really, really, really cool, I think. Let's see. Could you do a 3D reconstruction of an image from sampling the entirety of K-space? Really interesting idea. So each slice of the MRI, so the MRI actually is acquired in slices that is then post hoc reconstructed into a 3D volume. And these slices are actually like, there's a certain thickness of the slice that you are activating, and then there's a certain gap between each of the slices. So you can think about it like a bunch of stacks, but they're all slightly separated from one another. Each one of those stacks, that's an image that was created from a corresponding K-space image. Okay, so it's a slice and a K-space image, each one. The thing about K-space is that it's, well, there's two things. One is that, first of all, K-space is infinitely dense. So you can like sample to the nth degree as much of K-space as you want, as much of that frequency space as you want. But there's a bit of like a cost, opportunity cost, right? So you want to get as good of in the end, you want to get as good of an image as you want, which different sampling strategies will give you that kind of image. So the two, I'll just talk about just briefly touch on, I should actually put this in the slides. This would be cool. The two sampling strategies for K-space are, one is sort of like imagine K-space is a square, right? It starts at the bottom and then it goes across the entirety of K-space like this, in this kind of like zigzag pattern. So bottom, up, up, over, up, over, up, over, up, over. And it does that like pretty quickly. And then it goes to the next slice and does that for the rest of the slice. That sampling is then transformed into the actual, into the image. Another really popular one is called spiral. So it starts at the center and then spirals out. Another interesting thing about K-space is that it's symmetric. So you actually don't need to sample top and the bottom of the K-space, right? Because there's a symmetry there. So there's like sampling strategies that take advantage of that. There's all these really cool technologies and how to advance how this all works. So K-space is, you acquire an image from a slice. That slice has one corresponding K-space, and then the next slice has another one. Yeah. Okay, so where were we? Let's see, what were we talking about? Okay, now I'm going to get into actually an application of structural MRI. I just want to explain what these weird images are that I showed here. So like these two race car drivers are handing energy in the form of a hamburger between one another, right? But they can only hand the hamburger between one another because they're driving at the same speed. That's the idea with the radio frequency pulse rotating at the same frequency as the hydrogen atom is itself rotating. That's how you can successfully transfer that energy from one place to the next. And then the top one is just, it's harder to be in the higher energy state than it is to be in the lower energy state. Okay, structural MRI. So there's a lot of different ways to use this technology, to use MRI. I could speak to you guys a lot about all the different applications of MRI, but right now I'm going to focus on a couple of the key applications used in human neuroscience. So we have 3 techniques that I'm going to go over, and with these 3 techniques, you'll kind of have a good handle on 95% or more of all of the different techniques that we're using MRI for. There are many, many more that fit into that last 5%, but mostly people are like exploring whether they are valuable for investigation. The three techniques are structural MRI imaging, which I'm showing you an example of here, diffusion weighted imaging, and functional imaging. So the MRI signal that we're looking for in our studies is the one coming from the hydrogen atom, like I said. And since hydrogen is present in large amounts throughout our body and our brains, it's pretty much, and it's pretty much in most of the different molecules and chemicals that make up our brain, and all of the water that makes up our brain and bodies. And so, as we know, the signal for hydrogen shows up differently depending on what kind of molecule that hydrogen is in. And similarly, the signal that we get from the hydrogen is therefore different depending on what kind of tissue we're actually looking at. So we can tune the MRI magnetic fields in many different ways. And in this tuning, we can actually get very specific estimates of specific types of structures. So for example, if we want anatomical information about the brain's gray matter and white matter structures, we can use this kind of structural scan that's going to show us signals that are specific to those two types of brain tissues and that are going to differentiate between them. So liquids and fats have very different signal profiles. And so in structural scans, we get the gray matter coming out a little darker, and the white matter looks a little brighter. And since white matter has more fat, it looks brighter. And so in these images, though, it looks like the white matter is just these like big solid chunks, and the gray matter just wraps around it, but that's actually not at all how the actual brain really looks like. Actually, white matter is just packed, is very densely packed up fiber bundles. And you can think of white matter like these thick bundles of insulated wires that are following along like a main orientation. But the normal structural imaging that we do isn't actually sensitive to these fibers at all. So we use manipulations of the magnetic field in a different way to reveal the details of white matter structure, which we can call diffusion weighted imaging. So in diffusion weighted imaging, what we are looking at specifically is actually how water moves in the brain. So this technique takes advantage of this idea that when water molecules are allowed to move in an unconstrained motion, their average motion over time represents something like a sphere, right? And this idea is called like Brownian motion. So, the point of the water will move over time in something that looks like a sphere. It's probability density function. You can think about it, right? At time one, what is the probability that this water molecule will be in position A, B, or C, right? The probability falls off as you move from that point of origin for that particular water molecule. But when you put constraints on the direction of motion of the water molecule, then you find that the probability of where that water molecule will end up looking very similar to the constraints you put. So in the case of the brain where we're measuring water, we're measuring the water in and around all of these fiber bundles. We see that the orientations of these neurons places actually constraint on the motion of water within and around them, right? And so by watching how the water is diffusing in the brain, we can actually recreate very accurate maps of these white matter fiber bundles that fall in line with already existing detailed maps that were created by neuroanatomists of a previous generation. And the way that they did that is surgically very detailed with like post-mortem brains, very, very carefully finding all of these tracks with like a ton of dexterity. And a lot of that work has been done in Japan in like the 80s and 90s and stuff. And so by mapping, so now, now the cool thing is with this technique, the person doesn't have to die for us to be able to look at what these fiber bundles look like. So we can not only look at these fiber bundles in alive adults, but we can also look at them in kids and compare kids to adults and, all the different kinds of interesting questions you would want to do with living humans. So this you know, this sort of map of all of these different fiber bundles and where they're running, and this whole complete anatomical picture is what neuroscientists call the connectome, right? So the genome is sort of like an idea of how, you know, the map of basically all the base pairs of all of our genes, right? The connectome is kind of similar idea. Excuse me. It's kind of similar idea. It's the map of how the brain is fully wired up and connected. One of the downsides of this technique, unfortunately, is that it's actually really difficult to estimate the smaller scale architecture. So, for example, if I wanted to map between this point in my brain and this point in my brain with diffusion weighted imaging, I would be able to do that, right, from two sides of my forehead. But if I actually want to look at between two neighboring regions of the brain, the technology actually isn't really easily able to resolve that. So you can't really tell how well areas of cortex that are very close to one another are resolved. But nevertheless, we have this pretty decent large scale organization, sort of architecture, pretty well down to this point. And I am really interested in this idea of the connectome to understand the fundamentals of how the brain is organized, because to me, the idea of the connectome is fascinating, because the brain itself evolved as a network system to help the body move, right? To improve the evolutionary fitness of any animal that ended up creating a nervous system, right? So studying the brain in the sense of it being like a connectome, or like this, you know, multi-scale network, to me is really fascinating, because it feels like we're studying the brain in a way that it was really meant to be thought about. So this last technique that I'm going to talk about kind of takes that a step further. So you've probably heard of a functional MRI before. And so we can use this technique for a lot of really different and exciting purposes. And this is going to be the majority of what we're going to focus on today. But perhaps one of the most exciting elements of the functional MRI field is this idea of the functional connectome. So functional connectomics, I think, is maybe one of the most exciting new fields in human neuroscience, maybe more broadly in neuroscience overall. So diffusion weighted imaging gives us an understanding of like the underlying anatomical backbone of the connectome, but the functional MRI gives us an idea of how like actually that looks like in the end in terms of brain activity, right? So with fMRI and functional connectivity, we can have like a really good probe into how this connectome is producing things like cognition, emotion, learning, memory, and all the other, you know, fascinating aspects of the brain. Because we can not only look at the brain at rest, you know, when you're just kind of like laying in a scanner, but we can also look at the brain while a person's doing a particular task, right? And so we can kind of tease apart what sort of fluctuations in their intrinsic activity are related to that task that they're doing, like if it's an emotional processing task or a learning task, etc. So I'm I'm partial to this field because this is actually where I'm working. So you know, I'm fully acknowledging my biases here, and I'm mostly personally my work. I'm mostly interested in like creating a better understanding of the development of this functional connectome over childhood and adolescence, in particular how it relates to the development and the emergence of skills and cognitive development, and those cortical striatal circuits I was talking about earlier. So, okay, so if we take a look at the left figure, so we can see that the process, we can see the processes that these functional connectomes, by which they get figured out. So if we put someone in an MRI scanner, we just have them close their eyes. We just watch their brain activity. And we look at this brain activity in each of the little pixel of the 3D brain scan and assess how similar this pixel is with every other you know, pixel in terms of their brain activity. And this method is called functional connectivity, right? So we're calling things functionally connected if they have similar time series in their activity over time. And so by looking at the similarity of different brain regions, about 15 years ago, they discovered that there's actually a highly structured organization. This started about 15 years ago. And they discovered that there's a highly structured organization to the activity of all the regions in the brain. And we now started discussing them in terms of like major brain networks. So the figure to the right shows up at the top the most popular of the 7 network decomposition of the brain. And that's from a paper in 2011 by Thomas Yeo and Randy Buckner. Thomas Yeo is at Singapore now and is in Singapore. Randy Buckner is still at Harvard. So we have these 7 different networks. The motor network is in blue. That's responsible for movement and sensation. The visual network in dark purple, responsible for vision. The dorsal attention network in dark green, that's responsible for where things are in the visual field, like where they are in space. And the ventral attention network processing what things are in the visual field. And I want you to pay particular attention to this ventral attention network because this is sort of the part of the brain that we're going to be doing a lot of work into here, right? Because if you think about it, like the dorsal attention network is finding where things are in space, but the ventral attention network is actually where you get what we would call meaning from the things you're looking at, right? Like that's a cup, that's my wife, whatever. That's the part of the brain. So like that's what we're going to be doing in terms of decoding. We're going to be figuring out how the brain is actually processing that information, how it's representing that really high level information for you. We say these things like low level, high level to mean the difference between sensory and like meaning level, right? So for example, if I walk you through the visual field and I say, this is a low level stimulus, this is like a light, right? Like a point of light. As I move up the hierarchy of the visual system, eventually a sequence of points of lights will become a line. And then a sequence of lines will become a chair. And then eventually you'll understand through the ventral pathway that it's a chair. You'll understand what the thing actually is and what you can do in terms of your relationship with that thing, right? I can sit on it. So that's kind of like the low level, the high level dichotomy, if that makes sense. And let me know if you want me to explain that more, just feel free to ask a question.
Aki, you have a couple questions in the chat from Jamil.
Thank you. Yes, fMRI is measuring blood oxygenation level. So the type of fMRI that we are using is called blood oxygenation level dependent signal. So if you look at this bottom left sort of chart here, what you'll see is that the first series here is a stimulus, like a visual flash in the visual field, right? And then say we have like a little electrode put into the visual cortex, then we would have a spike in the response, right? Like you very quickly show the stimulus, and then the spike happens, but then when the stimulus is gone, the spike stops. And then you see that there is like a sort of delay. This flow response is talking about the movement of the blood into the area. And then this bold response, which is the thing that we study in fMRI, is delayed. So first thing, the stimulus and neural response happen almost simultaneously. And then the blood flow response happens a little bit after that. And then the bold response is kind of lagged and is both in start time and in eventual like end time. Bold is blood oxygenation level dependent. So we always talk about the bold signal. That's what we're always talking about in fMRI, the bold signal. In structural MRI, we're talking about the T1 signal or the T2 signal, et cetera. also more stuff I'm not gonna get into today. So the bold signal, so this kind of how the process works is that imagine now we're talking about stimulus, right? And there's a photoreceptor in my eye that is going to receive that stimulus. That photoreceptor is going, so I see the stimulus, it triggers the photoreceptor in my eye, that sends the signal all the way to the back of my brain where my visual cortex is, and where my lowest level sensory processing system of my visual cortex, called V1, is going to handle that point of light, right? The neurons in V1 fire. After they fire, the astrocytes, so astrocytes are like support cells for neurons. The astrocytes sense that the neurons are firing, and they send signals to the blood vessels, the nearby capillaries, to dilate. And when these endothelial cells dilate in the capillaries, more blood rushes into that area, right? But that process is delayed. So there'll be more blood after the fact, and generally kind of like it's a big response. So that kind of like over level of response is something that we're picking up in fMRI. And the fresh blood that's coming in has oxygen in it, right? And oxygen is paramagnetic and differently. So when blood's been, the oxygen in the blood has been used up, that blood versus the fresh blood has a different magnetic profile. This is what we are picking up on in fMRI. That's the blood oxygenation element of it. So we're not just picking up on how much blood there is, but the oxygenation of that blood in particular is the signal that we are picking up on. Okay. Which color is the ventral? The ventral is this light pink one. So the light pink is a pretty disperse. It's a pretty disperse one. It's like it's hard to see, but the reason it's called ventral is because it actually goes under the brain as well. So there's parts of it that are on the outside, right, or on this sort of area. You can see that like light pink, not the dark purple, but the lighter one. The ventral element of it is like underneath. So we don't see that rotation of the brain from this view. Just to be clear on what these brains are showing. So this is showing the like outside of the brain, and then the brain is symmetric, right? So it has these two hemispheres. This is showing the midline. This one down here is showing the midline. So the space, sort of the what it would look like if you just took out one hemisphere and we're just looking at it. That's what this is showing here. Okay. And then we have the limbic network in cream, like the cream color, mostly is also on the underside, so it's a little hard to see. That's mostly responsible for reward and motivation. And then finally, the default mode network, which is responsible for like internally oriented cognition and like, self type thoughts and things like that. And then the frontal parietal network in orange, responsible for executive function. And so the bottom right shows the same networks, but now they're broken up into individual regions that have similar activity patterns. And this is like a really important part of functional connectivity right now. This is the idea of parcellation and how to make parcellations, because it makes the whole problem of assessing the connectome a lot more statistically and computationally feasible. Because if you think about it, like 1,000 regions have like 1,000 times 1,000 divided by 2 number of connections, right? So it's like this big exponential. It's this big exponential growth. So if you have like 100,000 voxels, that means you have 100,000 times 100,000 number of connections, or divided by 2 number of connections between all of these voxels. So if you can get the number of regions down from 60,000 voxels to something more like 1,000 regions, then you can do a lot more. And as we will talk about later, with things like the curse of dimensionality, dimension reduction is a really important thing in any sort of statistical estimation. Okay, I think now we can open things up for questions for a little bit. And we're just hitting about at least for me, I'm hitting about the 1.5 hour mark. So maybe this might be a good time to take a little bathroom break before we jump into our first, the brain from the functional connectomics perspective, is that they are seeing that we have known that the brain has some kind of hierarchical structure for a long time, but it wasn't until kind of recently that we've started to see that hierarchy all in sort of one unified framework. So whereas before what we used to do is we would apply like techniques like independent components analysis, which would grab these like networks of the brain, but these networks were, what we knew is that these networks were separate from one another, but we didn't know necessarily how they are ordered in terms of their like hierarchy. And this new framework over the last few years has kind of emerged from some collaborators of ours at the Child Mind Institute, that is kind of taking what people call like a gradient approach. And so this idea with gradients is that it's kind of like similar actually to principal components analysis in some way. But what you can kind of see, for example, if we take this image C here, what we see is we see this first gradient kind of describes the transition of the brain from the visual system to the default mode system, and from the motor system to the default mode system. So how gradients work is they are based on the similarity of a region's connectivity to the rest of the brain. So instead of looking at, it's kind of like a one-step removed from the time series. So instead of looking at how two voxels in the brain are correlated to one another, what gradients are doing is they're saying how correlated are the connectivity profiles of these two voxels. So rather than so if I have like, it's a higher dimensional problem, which, as we'll talk about later, has some problems. How it's looking is what it's looking at is it's looking at characterizing how two voxels, if they're neighboring to one another, if they have similar connectivity profiles to the rest of the brain or different. And if they are different, then they sort of exist at a different part of this gradient spectrum. So this second gradient now here describes the transition from one main low-level sensory modality to a secondary main low-level sensory modality. So we'd like move along the gradient from the first gradient being hierarchical, and then the like second gradient being more of like a lateral move. And so now in this A figure, we're looking actually again at that that canonical 7 network organization, right? And what you'll see in these boxes is that you move repeatedly from this default mode network to through the higher order areas like the frontal parietal network, the ventral attention network, and the dorsal attention network, to eventually get to the lower level sensory areas. So it's like this. If you look in figure D, what you'll see is like the visual network moves through the dorsal attention network and the frontal parietal network to finally arrive at the default mode network. So there's this like repeated organization where the networks are stacked kind of next to each other. That has been kind of the first time we've started thinking about all of these hierarchical networks in one single model, right? So these gradients you can like characterize like the first 2 or 3 these gradients, and they just in those 3 gradients, they kind of capture the full like spectrum of the relationships between different brain networks. And one of the interesting things about these gradients is, so like if we take a look at this, if we take a look at the default mode network, so the default mode network is one of the most important networks of the brain, because people talk about it like the task negative network. So it's like it's the network that's active when you're not actively engaged in something, and it is responsible for like more metabolic demand than any of the other brain networks. So in kind of an interesting way, even though it's not strictly involved in a particular task, it actually costs a lot more for the brain to run the default mode network. So and it's kind of, as you can see, kind of sits in the center in some way of in this sort of gradient framework of all of these different brain networks. So if you take a look at each of these red spots, say in like figure A, and where they are, and then you start actually plotting the geodesic distance across the brain's inflated surface, what you're going to find is that like there are major like the major bound, first of all, these points are about equal distant from one another, right? So like the main points of the functional, like the hubs of these default mode network, these default mode network hubs, they are all like set up in the brain so that they're about equally spaced from one another. And you can see like the halfway points between them are actually mark some really important transition boundaries. So for example, like about halfway from the frontal DMN hub to the parietal DMN hub is this like strip of where the motor system lies, right? And so like with understanding actually where the main hubs of this like sort of central piece functional network is, we can actually start to explain some of why the brain is structurally organized in the way that it is, right? Because the brain is like very interested in being as efficient as possible with its use of like wiring resources. You could think about it in terms of like an electrical grid or something, right? You want to be able to convey as much information where it needs to go with the least amount of wiring cost. So you can kind of start to understand that, okay, well, we used to sort of think more like, oh, well, the brain are like, structurally organized like this, therefore the function follows from the structure. But in this case, we actually we don't necessarily understand that the function would be organized in this particular way with this default mode network. But then when we look at the default mode network, then we can kind of recapitulate how it's structurally organized, which is a pretty interesting thing. And if you are to look at the different parts of these gradients, and then like sort of look at which part of the brain or which type of visual like abilities or performative behavior associations they have, you'll see that as you move from one part of the gradient all the way to the other, so as you move from the dark blue to the dark red, you'll see this gradient that involves like an increasing order of complexity of the type of stimulus that brain region is responsive for. So like at the bottom of this gradient, we see like eye movements and visual perception and motor movement. And then as we go, we see like visual attention and multisensory processing. And then as we go up higher, we see working memory, declarative memory, autobiographical memory, emotion, and social cognition. So, this is kind of like I think that this framework is pretty interesting to sort of like holistically start thinking about the functional connectome, because, this 60,000 or 100,000 by 100,000 matrix that is the functional connectome that we are acquiring is pretty difficult to conceptualize, but then having these kinds of sort of frameworks helps us sort of think about it. Another area, another sort of area that's I think pretty cool that's been developed over the last several years is this sort of, and I'm going to get into this material more later, but this idea that like from the low level to the high level, we are like building meaning, right? So in this case, we're talking about, we're looking at these, and I'm gonna talk about this also in a second. We're looking at these like studies where people put people in an MRI. People put people in an MRI machine while they watch movies, and as the movie goes from scene to scene, what that you can actually see is that like the low level auditory information will be distinctly synchronized, matching up with the movie, with what the auditory stuff in the movie is saying. But like the higher level, like auditory material will sync up. Will take like larger chunks of the film, for example, to sync up. So like, you know, in this left part, right, we're seeing John and Mike talk in the park, right? And so from this multimodal region of the brain, it's synchronized over the whole length of this thing, because these high level or these higher level areas of the brain understand what's happening at this highest level. But as you go deep, like down into the sort of more and more detailed processing, you see that the synchrony is broken up into smaller and smaller pieces. So, okay, to like make this make more sense. So for example, let's take a look at this part of this film, right? And what we're going to see is, so this area right here, this is called the posterior cingulate cortex. This is like not only a hub of the default mode network, but it's also like an area for processing this kind of higher level information. You'll notice that it synchronizes in these large chunks, right? And it's synchronizing these large chunks because like the similar, it from a high level, basically the same thing is happening in the movie, right? It could be that, you know, the camera though is switching between one person and another person. So from the low level visual system, which is like represented by this dark purple part here, we see that the synchrony only stays for very brief periods of time, right? So if you can imagine like from part of the brain that's only processing this low level kind of information, it looks like it's seeing different stuff all the time. So it's not synchronizing up with itself over a long period of time. It's processing all that information really differently. But the higher level system understands what's going on more broadly in what's being perceived in the film. And so it synchronizes up over larger pieces of the movie. Okay. And you can also see that the off diagonals on average for the higher order systems are more different than they are. So what this means is like the processing, the signal profile of what's happening in this chunk of the scene versus this chunk of the scene. And there could be like some kind of scene transition in between this big chunk and this big chunk, in the same way that there's a scene transition between John and Mike in the park and Sherlock examines the body, right? Two very different type of things happening. You'll see that these higher order areas are very not, like they're not strongly correlated. They may even be negatively correlated with different types of scenes in another scene. Whereas the visual areas, they're strongly correlated with the time here, but if you look at other time points, they are showing the similar type of thing. And that could be as a result of like the low level stimuli, like, you know, both at, you know, say 26-minute mark, for example, that's a very bright shot. And then you go up here and this 27-minute mark, also a very bright shot. So both of those shots are registering similarly in the brain in terms of how it's perceiving that. Okay, and then I was going to do the Q&A, but we can skip over that given that we've already sort of had some of that Q&A time. We're going to have more time for Q&A as well. paper that we're going to talk about is this one, Distributed and Overlapping Representations of Faces and Objects in Ventral Temporal Cortex. So ventral temporal cortex, that's the part of the brain that we were discussing earlier. That is the sort of where visual attention network. And this study kind of showed a really interesting way to think about the brain that people hadn't really considered much before. So let me go back a bit. Yeah, so their MRI experiments, and I mean, they still do this, it's just like, this was a different way. The main way, when this came out, this paper came out, the main way that people would set up their MRI experiments would be, they would show a stimulus for a set period of time, like this block right here, and then they would stop showing the stimulus. And then they would show the stimuli again, and then they would stop. And they would do it for maybe like 30 seconds at a time, right? And then what they would do is they would look at this sort of a model of the expected bold response, like how the bold response would look if a voxel or a single region were to be involved in that task, right? And then what they do is they do a similar kind of thing that they do with the seed, with this seed to every other voxel, is they would take this model and then they would basically correlate it with every other part of the brain. And the parts of the brain that showed a very similar activity pattern to the expected one would be ones that they would say were involved in the task. And ones that showed dissimilar activity pattern were ones that were not involved in the task. But this method of, this method that Haxby proposed is really different, right? So instead of this like normal fMRI task, rest type paradigm, where you have someone in the MRI scanner, you're showing them some houses, you're showing them some faces, and then you're looking at where there is brain activity. In this one, we're asking kind of a different question. It's not where is the brain activity, but like, where does the brain activity contain information about what the person is seeing, right? How do we actually get to that type of information? So Haxby developed this in this 2001 paper. This was still a very rudimentary application of it, but he developed this thing called multi-voxel pattern analysis to address this issue. So what we're going to do in the like kind of overall today is we're actually going to build an MVPA pipeline from the ground up and use this MVPA framework kind of as a window to explore machine learning and to understand the brain. So I think first things first, let's pop open that Haxby Jupyter notebook, that first one. To guide you guys through how to do that,